Yeah, go ahead.
All right.
Thank you, Margaret, Kate, everyone else technically bringing this together. It's my pleasure to introduce our grand round speaker today, Dr. Amir Zarrinpar. Dr. Zarrinpar graduated from the MSTP program at UCSD with an MD PhD, where I was honored to be his associate dean. He completed his PhD at the Salk Institute under the mentorship of Dr. Edward Callaway on the topic of local functional input to neurons in the deep layers of the right visual cortex. Dr. Zarrinpar subsequently pursued his residency in internal medicine and fellowship in gastroenterology and hepatology in the physician scientist training program, also at UCSD. He then stayed on and joined the faculty at UCSD with clinical interests in obesity and metabolism. Dr. Zarrinpar is engaged in basic translational and clinical research on the role of the gut and the microbiome in obesity and metabolic disease. He is the PI in several grants, including an R01 from NHLBI, an R01 from the National Institute of Biomedical Imaging and Bioengineering, an I01 from the VA, and an R21 and a U01 from the National Cancer Institute. Earlier this year, he was selected to serve as the program director for the physician scientist training program here at UCSD. He serves as assistant program director for the UCSD medical science training program as well. He'll be speaking today on harnessing native gut microbes, a novel frontier in live bacterial therapeutics for chronic disease.
Thank you so much, Dr. Mendel, for that very kind introduction. It gives me great pleasure to give this talk. Trying to figure out how I advance, I guess through the keyboard here, my slides, but here are some disclosures. I will not be talking about any products from these companies during this talk. And I was asked to give a little bit of my background. I grew up in LA in Tehrangelis, one of the largest Persian communities outside of Iran. And really wanted to get out of there when I grew up. And I went all the way to the other side of the country, where I went to Harvard College. And there I really pursued my interest in neurosciences by joining the cognitive neuroscience lab of Stephen Koslin. He just looks like the prototypical Harvard professor. And I just thought that he had the most incredible career. And I decided to, so he really made me think about pursuing research. And as a result, I applied to MD PhD programs. I got off the wait list here at UCSD back in '99. I've been here almost 24 years. As Jess mentioned, I did my research in Ed Callaway's lab at the Salk Institute. Really, the most important lesson I learned from him was the importance of using technology to advance approaches to a biological question, and how by having a new technique, you can essentially corner the market when you're going up to answer scientific questions. Simrajot was my continuity clinic preceptor, and she was laying the foundations of perhaps going into internal medicine while I was with her. And I really enjoyed taking care of patients with her. I learned tremendously from her. And thus I started to consider internal medicine. One of the people that was really formative was John Crothers. He actually helped me realize that there was a need for neuroscience research in gastroenterology, which made me then pursue the PSTP with the hope of studying enteric nervous system or neurogastroenterology. Kim Barrett, who was the Dean of Graduate Studies back then, was also very formative in guiding me. And of course, one of the reasons why I decided to stay at UCSD was actually the near mentors. So Barbara Young had just finished her PSTP. Pradipta was a senior fellow at that time. And Will Go was a couple of years ahead of me, and just seeing how they went through the program really helped me understand the advantages of being in a physician scientist training program. While I entered, I really wanted to do neurogastroenterology, but very specific patient interaction made me completely change my career. And this was facing a patient who was obese in my clinic and seeing him regularly, seeing him try to lose weight, and just being incredulous that we had nothing to really offer him except bariatric surgery, which was not realistic for this individual since he did not have the social support for it. And which made me incredibly it made me feel incredibly powerless, and it made me feel angry for the patient. And it really stoked a fire in my belly to go back to the lab and to better understand this. And perhaps the only PI who would take on a postdoc that had no experience whatsoever in metabolism was Sachin Panda, who took me in at the Salk Institute and has been a wonderful mentor since. He really helped me also realize the importance of asking good gut check questions and challenging people's assumptions. So it's an exciting time to be a physician scientist. It's truly an honor to now be director of the program. And so hopefully you'll kind of get the feel of the joy that is in this career from the talk itself. But before I begin, let's begin to wonder, you guys have probably seen lots of papers out there about the gut microbiome, how it changes with things. And so why is it really important to measure the gut microbiome or study it all together? And really, most importantly, it seems like any physiological process that's going on seems to be affected by the microbiome if you look in the literature. But more importantly, why do we entrust such important physiological systems like our immune system, our metabolism? These are fundamental to our existence. Why would we entrust them to other organisms that are not us? And oftentimes the question that comes up in our lab is, who's the puppet and who's the puppet master? So if the microbiome knows how to manipulate these signals and get us to do things like prefer a certain food or act a certain way or evade our immune system or gain fat, then are they in control or are we in control? So I think that this is a very fundamental question as to what we're trying to study when we get to the microbiome. Are we just an ingenious way for bacteria, a specific kind of bacteria, to survive, or are we controlling the microbiome in specific ways? Well, the gut microbiome, it makes up half of what you see here, right? So we're one-to-one microbial to human cells. But really, what is more important is that the microbial genes are 1,000-fold higher. And we're estimated to have about 46,000 genes, 20,000 that are protein encoding. But with the microbiome, our genetic toolkit expands to 46 million. And not only is that important, but what's also important about it, and I'd like for all of us to think of the microbiome in this way, is that our genes, the 46,000, take millennia, take eons to adjust to new environmental changes. But our portable 46 million genes are constantly changing and constantly responding to our environmental needs and what we need in specific situations. So they're incredibly dynamic and they adapt. They can become better at what they do, or they can disappear altogether. And that's why I can go to Alaska and live off of blubber in an environment that has no water in the environment and be able to survive, and also on the same day be able to go to Hawaii and survive off of fish and in that warm tropical environment. That we have these genes, these bacteria that are expressing genes that we need, and they adapt to our environment around us. So today I'm going to first talk about something a little bit different, time-restricted feeding, and how this made us interested in the role of the gut microbiome and metabolism, and how basically questions that we could not answer led us to develop a new tool to help us understand what the bacteria in the gut were doing to affect time-restricted feeding, and then how we use this tool to address other chronic diseases. So first, let's talk about time-restricted feeding. So when I joined the lab in Sachin Panda's lab at the Salk Institute, we had just made the observation that any model of obesity that you had, any mouse model, involved the mice eating in abnormal times. So if you give a mouse a high-fat diet, it spreads its caloric intake to night and day. And by doing so, it also dampens its normal circadian genes and the circadian rhythms. It's awake when it's supposed to be sleeping. A lot of things are rhythmically going haywire. And of course, we see a lot of the consequences of this phenotype by noticing that they get fatter, they become diabetic, they have fatty liver disease, and so on. So really what we first wanted to determine was whether if you restrict access to food to the normal time when mice are supposed to eat, the nocturnal period, whether it can prevent a lot of these things. And so we did actually show that when mice are consuming all of their calories in the nocturnal period, they maintain these robust circadian and metabolic rhythms, and they get none of the consequences of this really bad diet. And we actually we have gone on to show this in many other diseases and metabolic conditions. Our colleagues here, Mike Wilkinson and Pam Taub, actually showed this in a cardiac population as well, that some of these metabolic benefits are maintained in humans as well when they do time-restricted feeding. And you know, this is probably the rise of a fad diet that you guys have noticed over the last 10 years. But one of the things that I noticed when we were looking at this data was that the microbiome itself was circadian. Back then, the assumption was that once you were weaned and you were in your particular culture, you had a specific kind of microbiome that you had for the rest of your life, and that it was very difficult to change this microbiome. However, the data that we were seeing from humans who had suddenly changed their diet, as well as the observations we were making in mice, was that there were these very vigorous fluctuations to the microbiome itself, and that these rhythms were disrupted by a bad diet, like a high-fat diet. So one of the big questions in circadian biology was that so first of all, we all have these timekeeping proteins in all of our cells that help coordinate many cellular and physiological processes across our body. But how does perceiving light with our eyeball and with our brain, how does that entrain all these different clocks in the rest of our body? And this was really an unknown question at the time that I was starting. And of course, when you don't know how something works, you blame the autonomic system. You say that perhaps there's some, weird hormone that we haven't discovered yet that's doing this. But really what the preponderance of evidence from research is showing us is that the way the brain can entrain the rest of the body is by controlling when we eat. And not only by controlling when we eat, you absolutely need the microbiome to do these entrainments. That if you take a germ-free mouse, that its whole circadian rhythms are messed up, specifically in metabolically important organs like the liver, muscle, fat, things of that sort. Whereas its brain rhythms tend to be normal because they're entrained by light. So this was a big clue in understanding what was yet another physiological process that the microbiome was affecting. And there have been a lot of theories as to what specific agents from the microbiome itself are these potential entraining agents that are giving these big environmental clues to our body and telling the body what time it is. And those are, of course, short-chain fatty acids, which are basically the breakdown of complex carbohydrates that can only be produced by bacteria, and then modified bile acids. So the bile acids, we're going to get into a little bit right now. Let me just go here. Bile acids are, you guys have probably learned in medical school that the liver makes bile acids to help us consume fats. Well, bile acids are, the way that they break down fats is that they're just like soap. They break up big fat globules into smaller and smaller ones. And of course, bacteria don't like being around soaps. It can disrupt their cell walls. It can affect their ability to survive. So they have developed different genetic tools to be able to survive in an environment where there are a lot of soaps like bile acids around. And one of the genes that they have is bile salt hydrolase, which deconjugates bile acids. It removes this amino acid, this attached to the bile acids takes it off and makes it a much weaker soap. And then once it becomes deconjugated, other bacteria can get at these bile acids and use them as carbon sources, energies, substrates, and so on and so forth. What's been really interesting is that all these different bile acids that are produced by bacteria are ligands for various bile acid receptors. They're either antagonists or agonists. And I've put two of these over here, FXR, which is a nuclear hormone receptor, first thought to be involved in cholesterol homeostasis and bile acids, but has now been found to affect glucose, lipid, insulin signaling, and affect the immune system, among a whole bunch of other things. FXR receptors are everywhere. And then TGR5, which is a G protein coupled receptor, which is much more poorly understood than FXR is, but it seems to be playing a huge role in various processes as well. We had noticed that in time-restricted feeding, that the variation in the conjugated to deconjugated bile acids changed with time. So in a normal mouse, you would see higher levels of unconjugated bile acids to conjugated ones in the daytime, lower ones in the dark, so on and so forth. Across all bile acids in normal mice, we saw this pattern. In diet-induced obesity, we noticed that this pattern shifted. They basically became the same at light and dark. And one of the things that time-restricted feeding did was that it restored these patterns. This was actually work done by one of the postdocs in my lab, Carolina Dantas Machado, and we recently published this. Another graduate student in my lab, Stephanie Flores Ramos, decided to look at the metatranscriptome. So usually when you hear about the microbiome, you're hearing a lot about what bacteria are there. What Steph did instead, what she was interested in, what genes are the bacteria expressing? And one of the things that she saw was that bile salt hydrolase, this gene that we know affects the levels of conjugated and deconjugated bile acids, was affected specifically in the light period of the mice in the time-restricted feeding condition. So that made us then think maybe these bile acid changes by the bacteria are doing something. Carolina went on to show that bile acid signaling pathways are different, that GLP-1 expression, which is one of the things that bile acids can control in the ileum, are affected. And in fact, we did show changes in GLP-1 and cholesterol. But so overall, what this is really telling us is that, part of the reason that diet-induced obesity is causing problems in the host, as we like to call it, is that there's a change in circadian fluctuations. Part of the benefit of time-restricted feeding is by restoring or maintaining these cyclical fluctuations, and that the microbiome has these circadian fluctuations that are vital to host circadian rhythms and part of normal homeostasis. And the bile acids could potentially be playing a particularly big role here. However, this is all very correlative. We're just seeing patterns change. We're not really able to manipulate these things to see whether they are actually the cause of the things that we're observing in the host. So that's what the next part of the talk is about, us trying to figure out, well, how do we manipulate what the microbiome does to affect host physiology? So we were left with a question of, are bile acid modifications the thing that the microbiome is doing that affects time-restricted feeding? How do we get past the correlation? And we want to knock in this function. So we are very new to this idea of engineering bacteria to do things, but what we had noticed in the literature was that there were many people who were using ever so creative and complex circuits to in engineered probiotics to affect the host's health, but these were primarily done in germ-free or reduced community, meaning mice that are bombarded with antibiotics kind of systems. You can't really study metabolism or perhaps even the immune system in these systems. So we really needed to figure out how to make this work in a real mouse with a real microbiome. And the way that the problem that we thought that was affecting this is that one of the things that our microbiome does is that it keeps other bacteria out. right? That's what we want our microbiome to do. If there's a pathogenic bacteria coming in, it muscles them out. Our bacteria is so well adapted to us that asking another bacteria that has never lived in the gut to come in and be able to survive is just not really realistic. And so, so a lot of the data we were seeing with these engineered probiotics, particularly the ones in humans, was that they're safe in the sense that they're not really doing anything, but they're not able to have these physiological effects. So we thought that the chassis is the problem. And when I say chassis, what I mean is that the bacteria that we're using to deliver the function is the problem here, that chassis is not able to survive. And in fact, this has been shown in a number of studies where one of the bacteria that people really like to use is E. coli Nissl or Lactococcus lactis. E. coli Nissl has been a probiotic since 1917, ever since it was isolated from a German soldier who did not get dysentery during World War I, and has been used as a probiotic since. L. lactis is a very common bacteria that's found in yogurt. So people were trying to engineer things that were safe already, but what we see is that these bacteria do not survive very long. In fact, they're barely detectable at 24 hours, and they're completely disappear in 48 hours. So we needed a chassis that was strong enough that can engraft and stay and live in the gut, survive and do the function, but easy to contain. And we also wanted a strategy that could eventually be translated to humans. If we create a really awesome probiotic or engineered probiotic that works in a mouse, that does not guarantee that strategy or that bacteria is going to work in humans. So we wanted something that would be easy to translate. The idea that we came up with, and this was work done by a gap year student, Bailey Russell, in my lab, who's now a PhD at Harvard, was that we decided that, well, why not pick a bacteria that's already well adapted to live in the gut? Let's isolate something that's native to the microbiome itself, and then engineer that bacteria to do the beneficial function that we want. This is kind of like CAR T, where you're using the immune system, you're using the patient's own immune system to fight cancer. Here, we're trying to see whether we can use the patient's own microbiome to express different functions and to understand what is going on. So, so when Bailey did this experiment, we showed that these bacteria, instead of surviving for just 24, 48 hours, they survive for the rest of the life of the mouse within the gut microbiome. You can see here that we followed this for 300 days. In this other experiment, we followed it for 2 years. We followed these bacteria and these mice for now generations. And not only does the bacteria survive, but it retains the function that we've engineered in it. And that function remains functional during this whole entire time. It doesn't lose its ability to convert bile acids. So, so we thought that this was a very exciting point that we were able to do this. The reason why there's this big gap here that we didn't check was because of COVID, by the way. So we decided to check one more time before finishing that experiment. So where do these bacteria engraft? Well, these bacteria primarily engraft in the cecum and ileum, but you can find them almost anywhere along the gut. And this also was a big change from a lot of the probiotics. So even when you do find your probiotic after 24 hours, it's primarily in the colon. But now that we can get engraftment in the small intestine, it can really affect a number of diseases or potential things that we can affect. There was always concern from our vivarium whether this bacteria can spread from mouse to mouse. And certainly we didn't want that to happen. But so we decided to do an experiment where we took 2 mice that were living together in the same cage, and we put the bacteria in one mouse. The other mouse did not have the bacteria. It just got a vehicle gavage. And then every week we would go and check for our bacteria in both mice. So on the second time that we took these measurements, we noticed that we were detecting the bacteria in the cage mate and the one that did not receive the bacteria at the beginning of the experiment. We were kind of bummed. We were like, oh man, it's really spreading fast. But then we thought, well, we're detecting DNA. is this just digested DNA, or is the bacteria actually in the gut of this mouse? So we made a small modification in the experiment where the day before we were going to measure for a bacteria, we just separated the mice for a little bit, for 24 hours. And after we did our collection, we would put the mice back together again. When we made that change, we see that the bacteria actually does not engraft in the cage mate. So even though this mouse is, since mice like to eat each other's poop, even this mouse is consuming this bacteria, it is not able to engraft. And so it doesn't pass between cage mates. And we're like, well, how is that possible? How can we get it into one mouse by gavaging it in, but the other mouse doesn't get it? Well, it seems as though you need a critical load of this bacteria first to get in to be able to engraft. And that critical load just does not exist in the amount that you find in each other's poop. So, so these mice do not spread the bacteria between them, which is really cool because you can do experiments where your control and experimental condition are in the same cage, they're brethren, everything is exactly identical. The only thing that's different is one gene in one bacteria. So, so I showed you this before as to what bile salt hydrolase does. One of the questions, so one of the things actually about bile acids is that we're really, really good at transporting. We have actual transporters for conjugated bile acids. We want them back. We want those soaps back. We want to recycle them. We want to use them for digesting other lipids. The deconjugated ones, they have to go through passive diffusion. We don't have a really good way of collecting those back, and that's why they end up in the colon and get converted into secondary bile acids. So what we first expected to see is that since we thought, well, there's going to be a lot more conversion of conjugated to deconjugated bile acids, we would expect the amount of bile acids in the poop to go up. And that's exactly what we saw. And just to have people like understand what they're looking at here. So this is a mouse that we treated, and then we came back 3 months later to measure these things because the bacteria is engrafted in there and it's doing this all the time. So we did see an increase in the amount of fecal bile acids. We saw a decrease in the conjugated bile acids. So the amount here was decreased in these mice that had increased bile salt hydrolase activity. We see an increase in these secondary bile acids, which is what you expect since it's going down this pathway more. And then more importantly, the bile acids that are specific antagonists and agonists of the receptors that I just mentioned, those are highly affected by this bacteria. So we thought, okay, we're seeing all these changes in bile acids. We must be impacting the microbiome itself in some way, but we're not. The microbiome itself is indistinguishable between any of our mice, whether they're treated with the bacteria or not. And that's mainly because this bacteria that we're using, this E. coli that we isolated from the mice themselves, comprises less than 0.1% of the bacteria of the microbiome. So there's no way of really distinguishing between these mice based on the microbiome alone. And we thought, oh, we noticed that the mice were more prevalent in the ileum and cecum. So we thought if we tested there to look for microbiome differences, we'd see some difference. But we didn't see anything. And this actually blew the mind of our reviewers as well, that there was no distinguishable pattern. So we're not the first people to put bile salt hydrolase into mice. Other people who've used these engineered probiotics have put them into germ-free mice or in mice that are heavily treated with antibiotics. They've shown that bile salt hydrolase can cause weight loss and can affect insulin sensitivity. We did not notice any change in weight, and we were really worried that the experiment was not working, that these mice, even though they were engrafted, even though we were seeing these bile acid changes, that it was not enough to create a physiological change. But we did see some promising results with postprandial glucose and postprandial insulin. So in both of these cases, the mice that had received the bacteria with bile salt hydrolase were protected against these things. And then those were just normal mice and normal chow diet. What if we put it in a diabetes model? So we took these mice that are the OBOB mice. These are mice that have problems with their leptin receptors and actually their leptin production. And what happens is that they just are eating all the time, eating. And that's how they become fat and diabetic. When we give this bacteria to OBOB mice, they're still eating a lot, they're still fat, but 15 weeks after we gave them the bacteria, we come back and check their glucose tolerance. They have an improvement in their response. So that was incredibly encouraging news that this function is actually doing something to affect host metabolism. So, but everything I told you came from a circadian study. So what if we apply this to an animal that has circadian dysfunction? And one of the benefits of being a junior faculty member is that you are a candidate for a lot of random grants. And there was this random grant that was from the American Federation of Aging Research that was for people who did not do aging research in the hope that they would do aging research. So, so we decided to look at aged mice. And in aged mice, what you see is that mice tend to be way more aged mice, tend to have more body fat. They have poor insulin, they have a lot of insulin resistance actually. And this happens actually in context of them not eating that much. And one of the things that we observed was that just like the diet-induced obesity mice, these aged mice have perturbed eating patterns. So they're eating more calories in the light period when mice should be sleeping than in the dark period. We also found that their bile acids were affected, their microbiome dynamics, so on and so forth. So one of the postdocs in my lab who started this project, and Carolina, Anand Saran, who started it, and Carolina, who's continuing it, decided to take mice that were already aged, that were already phenotyped. And what if we took these mice and treated them with our bacteria that had either bile salt hydrolase or not, and looked at these metabolic markers? Well, one of the things that we noticed was that these mice started to lose fat mass, but not lean mass. And in fact, even though their weights was not significantly different, they tended to have a lot less adipose tissue. Oops, sorry, clicked the wrong button. And their insulin, their insulin sensitivity improved as well with this bacteria. But perhaps what was most surprising to us is that they changed how they ate. And aged mice that had received the bacteria with bile salt hydrolase were now consuming more of their calories in the dark period compared to the light period. And not only that, one way that you can measure sleep in mice is that you can just look to see how active they are in the cage. We see that the mice that had received bile salt hydrolase bacteria were just less active in the light period and hence were sleeping more. And because they're sleeping more, we think what's happening is that they're actually using their fat stores. So we see a decrease in the respiratory exchange rate. The lower it is, the more fat they're using as a carbon source. So, so we think that this is how this metabolic benefit is occurring in these mice. So I showed you that engineered native bacteria allow for perpetual colonization of conventionally raised hosts, and that we can functionally manipulate the gut microbiome host physiology, that a single gene in a single bacteria that comprises less than 0.1% of the microbiome can have such huge metabolic effects. I say that and people often say, how could that be possible? But we already know this. If this bacteria had if this E. coli had a pathogenic gene, it can definitely affect the whole physiology of the mouse. So now we're just giving it a beneficial gene. And that bile acid modifications can replicate aspects of time-restricted feeding, particularly when it comes to insulin sensitivity and glucose regulation and feeding behavior. So now we have a way of knocking in functions into the microbiome. Why don't we just go and see what else we can treat with this, right? That was the thinking of the lab. So, so can we treat other diseases? And we've actually took this way too far, but I'm going to show you data for two different major projects in the lab. One is colorectal cancer, which remains the third most commonly diagnosed cancer and third most common killer in cancers. And we were really encouraged by this paper from Ron Evans' lab at the Salk Institute that showed that this bile acid receptor, FXR, is a tumor suppressor. And we thought that our bacteria was actually activating FXR by removing its main antagonist. And so we thought, hey, we could potentially have a cancer suppressing gene. But then another paper came out that showed that this other bile acid that we're increasing, deoxycholic acid, could be causing DNA damage and making cancer worse. These are the best projects. Either way, you're going to find something, right? Like it's either going to, like you can either say that the microbiome makes cancer worse or better. So we decided to do this and to see whether these bile acid modifications can promote cancer. This was work done by Ariana Brevi in my lab. We graciously received, this is part of the benefits of being in the home institution. We graciously received some mice from Pradipta Ghosh's lab to start this project. And what we noticed actually was, again, very disappointing. It's like what we saw was that some of the mice that we had engrafted with our bacteria has started to lose the bacteria. And we had never seen that before in hundreds of mice across multiple years. But we just decided to continue and see what happens. And what we noticed was that some of our mice were cancer-free. Not a single evidence of cancer in those mice at all. And all of these mice that were cancer-free had lost colonization of the bacteria. So, so we don't think that this bacteria is affecting cancer through bile acids. We think it's somehow making the immune system aware that these mice are developing adenomas. So that's how we're pursuing this further. There's a lot more work that needs to be done there. We've also started some exciting collaborations with colleagues at the University of Florida who are helping us design other types of bacteria that can provide cytotoxic agents or detect the tumor itself. And then another project that we decided to pursue was PCOS. For those, many of you are clinicians, so you know that it affects a lot of women. There's elevated androgens, ovarian cysts, insulin resistance, and is often goes undiagnosed until the patient wants to conceive. There's no treatment to reverse PCOS. Birth control or ovulation stimulating drugs can help patients control their symptoms and get to conception. Weight loss can be effective as a treatment, but there are a lot of women who develop insulin resistance, develop PCOS, but are not obese. So there's a really good animal model for PCOS. There's actually a bunch, but I'll be talking about one specifically, which is letrozole administration. This is actually, we received a lot of encouragement and support from Kana Thackeray from who helped us get this started. And she had done some experiments showing the importance of the microbiome in protecting against PCOS. Again, showing that there were some bile acid changes occurring with these, but again, we did not have a way to actually test to see whether these bile acid changes are responsible for the benefits. So we decided to team up and see whether our bile acid modifying bacteria could affect PCOS. One of the first things we did was we went, most diabetes experiments in mice are done in male mice. We went to see whether androgens in these male mice were affected. And to our surprise, we noticed that all the mice that received bile salt hydrolase had lower levels of testosterone. This was across multiple studies. It was repeatable. So we thought, well, it either is going to reduce androgens in these mice and protect them against PCOS, or it's going to reduce insulin resistance because we've seen that in our male mice as well. So it's probably going to be beneficial. And sure enough, actually, I should say, so Erica Mazie in my lab, a graduate student in my lab, did this project. And sure enough, she found that the mice in our PCOS model that were treated with our BSH-containing bacteria had a greatly improved insulin sensitivity. But what was surprising was that mice that are receiving letrozole often bulk up because they're essentially getting an androgen and they're building muscle mass. But what we were seeing in these mice was a decrease in muscle mass. So they're getting improved insulin signaling in the setting of decreased muscle mass, which is not something you usually see or even think about. So, and then of course their ovarian weight went down as well. We're getting the histology results for that right now. So potentially we can affect tumorigenesis with these bacteria. We're hoping that this will have an impact on patients with FAP or other hereditary causes of colorectal cancer. This is really telling us that chalk another thing, again, who's the puppet master? The microbiome is able to control reproduction. And I think that in itself is very conceptually interesting as to why would we allow, as hosts, allow another organism to control. So my final slide is basically, what are we doing in my lab? Well, we want to understand what microbial functions are out there that could potentially affect host health, and to use this system to study the effects of bacterial functions. The microbiome is very dynamic, and I can tell you right now from our studies that our antennas for detecting changes in the microbiome are only up for certain times of the day. They're not up all 24 hours. So certain functions being performed at certain times are going to be very important. And then also investigating microbial bile acids as signaling molecules. We've been working with Peter Dorstein on this, since he's been able to characterize a lot of novel bacterial bile acids. And then also, one of the benefits of being a physician scientist is that you can start thinking about how can we use this as potential diagnostic tools as well to improve the way that we treat patients. So we do want to make microbiome-mediated therapies with curative intent. The fact that so many organizations that represent patients are insisting that we go after cures now has been very helpful, and we have over 25 active projects. One of the criticisms I get is the lack of focus in the lab, but we just think that it's really amazing to be able to look at the role of microbiome in so many different diseases. We've been fortunate to have support from a lot of different organizations for this work, but really this work is done by the people who you see here. And one of the best things about UCSD is the generosity of colleagues and the collaborators who are willing to go after these high-risk, high-reward projects with you. So I can't name every collaborator here, but this one of the best things about this institution. So thank you very much.
Why don't we start with the in-house questions here? Sure. Tool.
Yeah, that was great. I really enjoyed that. Two questions, if I may. The first is, some of the human data I've seen on time-restricted feeding has been somewhat disappointing. And I'm wondering if you think just the right study hasn't been done, or not looking at the right studies, or that there's some disconnect between the preclinical stuff.
Yeah, so the question was about time-restricted feeding in humans and how it does not have as much of a physiological effect in humans as it does in mice. And I think that there's a reason why I haven't been doing the human studies. I'm not as brave as Mike and Pam have been, as well as others in the field. And it's mainly because I think that it's not clear what the intervention is. So is the intervention to control for the amount of calories that people take? Because the studies that find that the effects are not as pronounced are also showing that time-restricted feeding is causing caloric restriction. And the controls are often other individuals with similar kind of caloric restriction. So I think that has been one of the challenges of doing human studies as to what's the appropriate control, what's the appropriate intervention. There's this debate as to whether early versus late time-restricted feeding and actual length of time. So I think that is part of it. But putting on my clinician hat, a lot of people are obese, and people become obese for different reasons. And I think that if you appropriately phenotype the patient, so if the patient is a shift worker, or the patient, when you discuss with them that they are spreading their caloric intake, it is much more likely that an intervention like time-restricted feeding will have an outsized metabolic effect in that individual as opposed to all comers. And I think that that's what Sachin is doing with the Healthy Heroes Project, which so he is investigating San Diego firefighters who have 24-hour shifts and seeing the impact of time-restricted feeding on that population specifically. Having said all that, mice and humans are different. One of the things that humans do to prevent loss of weight is we drop our resting metabolic rate. We can drop that by 50%, 75% to help us retain calories and not lose fat mass. Mice don't have that compensatory mechanism, which is part of the reason why we see 30% weight loss difference with time-restricted feeding, but in humans, we see more like 5%. So I think that there's a little bit of both in terms of
Another question.
Sure.
So American endocrinologist, which gives me enough knowledge to be dangerous. As I understand it, in polycystic ovary, there's insulin resistance in most tissues, but there's still sensitivity of the ovary, which is why they use androgen. So the improvement in insulin sensitivity you showed us, was that organ specific or get a sense?
We have to figure that out. That's on the list of to-dos, and to also determine. So what's interesting is that just like with any ligand for nuclear hormone receptors, bile acids and these hormones are very similar structurally. And there have been studies that show that there's a lot of, there's potential activity of bile acids on these receptors. So it's going to be hard to tweak that out. But yeah, so I think we're going to have our work cut out for us. I think the fact that the bacteria can modulate what we're seeing is in itself an important concept.
The androgen levels are.
The androgen levels are not going up in those mice now.
We're very curious about your dinner table conversations after that last question. Praditha.
Not microbiome.
I'll try to be loud enough, I don't know. So thank you for educating us on this. My question has to do with, do bacteria outside the gut, if you took it out, put it in a petri dish, do they have a cycle? The reason I ask is you brought up who is puppet master. We do know that our immune cells, though, taken out of the body, have intrinsic circadian rhythm. I don't know if it has been established the same for bug inside the gut, outside in the petri dish, as a population.
So, so the question was whether bacteria have intrinsic cycling or a way of keeping time outside of the host. That is a really important question as to whether the bacteria have any capability of anticipatory behavior, let alone time behavior. What we do know is that bacteria that do not live in the gut, some of them do have this capability. So they can anticipate when nutrients will be around. They can either to change their transcriptomic machinery. They can also, some of them detect light. So single-celled organisms do have the capability of having circadian cycling. We have not yet seen whether these machineries exist in the microbiome through metatranscriptomics. It may be that there are genes that we don't even know that are there. When you take these bacteria out of the gut, we do not see them have, at least the ones that have thus far been isolated and studied, there's no evidence that they have intrinsic cycles. So we are potentially the puppet master, but and the thing that actually, I guess that's if you want to talk about puppet master, we are absolutely the puppet master. And the way to think about it is that we are cultivating the same way that ants can cultivate fungi for nutrition. We are cultivating the right microbes to have at the right time. And why is bile acid such an important signaling molecule? I'm convinced that it's because our liver has no way of knowing how much fat we consume without the bile acids. So we know how much sugars, how much simple carbohydrates we're taking because of the sugars that come in. We know how much protein we're eating because of the amino acids that come in. But a lot of the fats go into the lymphatics and they don't go to the liver. So the liver knows what the fat consumption is based on the bile acids it gets back. And if you look at the strategy that the host is taking, decreasing number of pups potentially, activating the immune system, being a little bit more stressed, making insulin more sensitive. These are the kind of things an organism that thinks it's in a nutrient poor state would do. So I think that these are environmental signals, and we are trying to figure out what's going on in the world based on what we're seeing with our gut.
Yes, Paul.
So Amir, that was a really excellent talk, and wonderful project. And I was struck at the end, you said that you looked at 29 different clinical conditions. What do you see as the path for translating this into clinical treatment? One can imagine a lot of things, but I'm curious what you think of the resistance to that. What's the problem with the environment you immediately moving into that you've got already?
So the question is, why isn't this going to clinic already? Like, what are the barriers? Is that correct? In part. Okay. So yeah, so I think that there is we're in untrodden territories where this is not a small molecule. This is a permanent change. We are trying to figure out how to reverse it once it's there. We still haven't figured that out yet. That's why inborn errors in metabolism are there, because those are the people you never want to reverse these functions in. And I think that I think the FDA is still trying to figure out what a microbiome therapeutic would look like. And the fact that they approved 2 new microbiome drugs is very encouraging, mainly because those at our Digestive Disease Week a couple of weeks ago now, both groups showed that the way that their drug works is in engraftment, and the patients who do not respond to their bacterial treatments are people who do not get engraftment. So I think that I think the goal here should be to cause permanent change in the microbiome. I think we're still hazy as to what the full consequences of something like that will be, and what does it mean to reverse it as well. So, but I think that these are, I think that the FDA is very encouraging. I think that the path is just not clear, and so there's some skittishness with ventures and things of that sort. I mean, look at our funding. The FDA the NIH wants this. The NIH wants us to figure out mechanistic what's going on. But also, I will also pose another thing to you, Paul, which is that everyone thinks that a microbiome therapeutic will be a bacteria or maybe an engineered bacteria. But really, the best microbiome therapeutic is knowing what levers that bacteria presses and acting on that specifically. I think that is going to be, because a person's response to bacteria will be very different based on what microbiome they have, what diets they're taking, whether they have to take antibiotics or not. All these things will affect the behavior of that bacteria. But if we know what that bacteria is doing, the 20 years down the line is actually producing that product, causing that change in a much more reproducible and fashion that we know.
Final question.
I found your the key case not being able to pick up the engineered bacteria fascinating. I wonder if you looked at cups or covers being divided with those engineered bacteria being transmitted, and then even if they are being transmitted, I think those cups may still have some behavioral and metabolic effects that you see in another.
So the question was, is the bacteria being transmitted vertically between mothers and pups, and whether the phenotype transfers with bacteria? We've only tested the bacteria without the bosalt hydrolase gene for vertical transmission, and we've now followed it for 6 generations. So it's 100% transmission between mother and pups, and yeah, so it's just there. It becomes part of it.
Oh, because it wasn't eating the food, right? So that's actually my question. Thank you. How do you think that
I think it's because they do, either because they're in this nest that's full of feces and it gets smeared and they're suckling. I have no idea. Yeah, the question was, how does it get transmitted? But it's 100% transmission.
That's fascinating. They have multi-generational applications.
Yeah, and this gets to the concept that we think that the bacteria might have what I'd like to call epigenetic phenomena. So a mother is passing on not only her genes, but her microbial genes as well. And if those microbial genes get shut on and off by diet or stress or inflammation or whatnot, even though the mother's genes are not affected, we think that we can affect the phenotype of response of the pup based on what bacterial genes get turned on and off.
Okay, I think we have reached our time. I apologize. We did not get to all the questions. Amir, thank you again.
Thank you so much.
Terrific.
Thank you
and
for having me.
thought-provoking talk.
Yeah, I'm happy to answer them.
Okay.
Yeah, I have some time.
I'll copy them and then send it to you if you want.
Get to safety issues.
Okay, don't end the meeting yet, okay?
I'm not sure if I can end the meeting,
Individuals
actually.
came together, merge.
You're the host.
In terms of it, I mean, it's like, think of that experiment.
How's this? I'm gonna, I need to do something real quick.
Yeah, you know, that's a really good question.